proposed to take some author, which I was working on, and to tell you something about it. Thankfully, I have an article where I'm a co-author. It's called Targeted Delivery of Drugs Using Platinum Polymer Nanoparticles. What? Yes, it's a bit too complicated, but I will try to translate this into our normal language through my speech and explain why it is really important, and I was passionate to study this through several years in my university. So we all know that cancer is a very hard disease. It is a hard to cure disease, it's dangerous, it's even deadly. But why is it so? It is because of the nature of the cancer itself and because of the ways it is being treated. When one particular cell in an organism starts to change its program and it starts to divide itself all the time, it starts to reproduce rapidly. It reproduces then two new cells also reproduce into 4 and more and more and more, and this mass of cells that keep on reproducing, they form the tumor. And even if one cell will stay inside of the body, it wouldn't be removed, the same process will start all over again. That's why it's so hard to cure cancer. Two major ways of curing cancer right now is surgical removal of the tumor of these cells that keep on reproducing, and also combining with chemotherapy. Chemotherapy is some particular substances, drugs, that are very toxic. And we all know these side effects that are followed closely with these therapies. And if after the surgical removal and chemotherapy, at least one cell will survive, it can start all over again. That's why it's so difficult to cure. What if we could cure cancer simply by delivering these drugs to the cells that needs to be killed, to cells that are damaged, and leaving all normal cells alone and without damaging them? What if we could deliver these drugs to its target, tumor cell? Targeted delivery. That's what it is. My title is Targeted Delivery. What we studied in our work was a targeted delivery of chemotherapy drugs using nanoparticles. Now, what is nanoparticles? Nanoparticles are really cool things. They are being studied all over the world. They can be of different shapes, different sizes, though they are very small, like few molecules. They can be out of different materials, out of metal, silver, gold, polymers. They can do different things and they have different properties. Such nanoparticles can be loaded with chemotherapy drugs. That means that we have our nanoparticles and connect to it our substance that is used in chemotherapy. But until the nanoparticle reaches its goal, tumor cell, it won't release it, so normal cells will stay alive. And also we can use some specific other molecules that will recognize specifically the cell that we need, tumor cell. That's how the targeted delivery of drugs can be done. Then, when a nanoparticle comes to its target, to a cell, it recognizes it, and then it can release the drug close to the cell, somewhere near to it, or it can go inside of the cell if the cell is able to capture some particles, and then the drug will be released inside of the cell, even not being somewhere in the blood flow. That makes it extremely efficient, because all of these high doses of toxic substances will go strictly to the tumor. What we've done in our investigation is testing is using macrophages. Macrophages are the cells that can capture nanoparticles. And macrophages can be bad when blood cancer starts. So blood cancer influences these kind of cells, which are called macrophages. So we used, this is a new photo from our study. We tested on them our nanoparticles. At first, we needed to make sure that nanoparticles that we need to use will actually come inside the cell, that they're big or small enough to go there. And we tested them, used a test for visualization. We stained them with some particular paint, let's say it, and we really showed that here you can see these green circles are cells without nanoparticles. They're simply green, and with nanoparticles, these orange spots are actually the nanoparticles that came inside of the cell. So we knew that they can be captured by cells. Then we loaded them with chemotherapy drug and started to put different amounts of nanoparticles to cells. And we've seen some statistics that the more we use nanoparticles, the more we put them. This graph shows that the more we use nanoparticles, the less living cell stays in some specimen. So if we use more nanoparticles with this drug, it means it will be more and more efficient. Is that clear? Maybe any? Okay. This is actually a promising result, and we're passionate to continue this work, to investigate it more. And even if we won't have this solution, we really hope that somebody else in the world, in other laboratory, will find this combination of nanoparticle and chemotherapy drug that will actually solve the problem of human cancer. Imagine that cancer could be cured like a flu. A person takes like 2 pills in several weeks, and he has no side effects, he even don't think about it, and he's just healed. That's why we're working on it. And if you have any additional questions, you can come to me after the meeting. And now